friends, today we will discuss about factors responsible for drug receptor and interactions, dose response relationships, drug potency and efficacy, and therapeutic index. And our main objectives will be: first, introduction about drug receptor interactions; second will be to know about dose response relationship and to get knowledge about drug potency. The drug's binding to specific sites or receptors was first discovered by Paul Ehrlich while studying the interaction of dyes with biological structures, which was later revived by Drews. Receptor is a macromolecule or a binding site located on the surface or inside the effector cell that serves to recognize the signal molecule and initiate the response to it. Ehrlich initially believed that receptors existed only for toxins and for physiological foodstuffs and ferments. He changed his mind only in 1907, partly due to results of his own further research, but in particular also because of a different kind of receptor theory that have been proposed by the Cambridge physiologist, that is John Newport Langley. Langley's concept is particularly interesting because it places special importance on the notion of substance and of substance binding. Receptor theory was propounded by Alfred Joseph Clarke, a theory of drug action based on occupation of receptors by specific drugs, and the cellular function can be altered by interaction of the receptors with the drugs. The interaction between the drug, that is D, and receptor, that is R, is governed by the law of action. The rate at which new DR complexes are formed is proportional to the concentration of D, that is drug. This equation is derived from Langmuir absorption isotherm. The interaction of drug with receptor on forward or association rate constant and the reverse are dissociation. Drugs target receptors because receptors are involved in chemical signaling between and within cells. Receptors can be located on the cell membrane, within the cytoplasm of the cell, or on cell nuclei. Activated receptors regulate cellular biochemical processes either directly, for example, causing a channel to open and allowing ions to flow through, or indirectly, for example, causing a cascade of events in a cell that eventually causes protein production. The molecules, for example, drugs, supplements, chemicals, hormones, and neurotransmitters or peptides that bind to receptors are called ligands. Ligands bind to specific regions of receptors called recognition sites. The binding site for an exogenous, that is not present in your body, drugs may be the same as or different from endogenous, that is present in your body. Ligands, for example, hormones or neurotransmitter. A ligand that activates a receptor is called an agonist. The activation of receptors can increase or decrease or inhibit particular cell functions that are already ongoing, or the ligand can start a cell processes from the beginning. A ligand that blocks a receptor is called an antagonist. Many ligands can interact with many different subtypes of receptors. This degree to which a drug acts at a particular receptor site related to other sites is called ligand specificity. A drug's affinity for a receptor is a measure of how tightly the drug binds to the receptor. The intrinsic efficacy of a drug is the degree to which the drug activates receptors. Some activate it maximally, some only partially. Both the affinity and efficacy can contribute to the degree of length of biological response produced by a drug and are influenced by the chemical structure of the ligand. The pharmacological effect of a drug is also determined by the duration of time that the drug-receptor interaction lasts, that is called residence time. Residence time is affected by dynamic processes of the receptor conformation change that control the rate of drug association and dissociation. A longer residence time can explain a longer pharmacological effect. However, a longer residence time can be a disadvantage because it can also lengthen a drug's toxicity. The ability of a drug ligand to bind to a receptor is influenced by external and environmental factors as well as by intracellular regulatory mechanisms. The amount of receptors present at the site of action and the efficiency of response varies from tissue to tissue. The presence of other drugs or supplements or foods, aging, genetic mutations and disorders can increase or upregulate or decrease or downregulate the number and binding affinity of receptors. For example, excessive caffeine intake upregulates adenosine receptors and causes tolerance to the effects of the drug. Chronic therapy with beta blockers upregulates beta receptor density and severe hypertension or tachycardia can result from abrupt withdrawal. There are three forms of binding to receptors. First is agonists, second antagonists, third is partial agonists. Now coming to the ligands. Ligands are the endogenous substances, molecules or compounds which bind with receptors present in the body. For example, acetylcholine, adrenaline, non-adrenaline or neurotransmitters like glutamate, aspartate and GABA. They produce various effects and interfere with the inflow of ions through channels called ligand-gated channels. Their action may resemble with natural ligand, block the natural ligand. Now coming to agonists. Agonists are the drugs which when bind receptors cause activation of receptors. They have the capacity to produce chain reactions in the receptors which ultimately bring about the effects. Agonists have two basic properties. First, affinity for receptor, second, capability to produce chain reactions in the cells having capability of intrinsic activity or efficacy. Now coming to the coupling. Transduction processes between occupancy of the receptor by agonist and response to occur is called coupling. Most agonists when bind to receptors cause activation. Most of them alter the second messenger systems in the cells. There are three messenger systems. First is cyclic AMP, second is cyclic GMP and third one is calcium and phosphoinositol second messenger system. The levels of these second messenger systems may increase or decrease. This may occur in three steps. First is drug-bind receptors. Second is stimulation of G regulatory protein occurs. G regulatory protein exists in two forms, that is GS, that is stimulatory G protein, and GI, that is inhibitory G protein. And third one is this causes change in the effector element, enzyme or ionic channel depending on GS or GI. When drug binds to the receptor, activation occurs and the response gradually increases until it reaches the peak. Then the response decreases, although agonist still binds. This is due to the desensitization of receptors, which might be overcome by removing the agonist from the receptor. Example includes succinylcholine, that is succinylcholine, which produces relaxation of skeletal muscles by activation of nicotinic receptors or desensitization of receptors in the neuromuscular junction. The reason for desensitization is not clear. In beta blockers, which act by CAMP, ultimately phosphorylation of proteins and accumulation of phosphosylines occurs, which increases the binding of beta receptors with proteins, known as beta-arrestin. This beta-arrestin interferes with the activity, ultimately leading to inhibition of adenyl cyclase enzyme. The binding of drug with the receptor may be of two types. First, it may be reversible binding and second, it may be irreversible binding. In reversible binding, the bond between the drug and receptor is very weak, ionic. hydrogen or van der Waals interaction. This effect is short-lived in irreversible binding. Very strong covalent bonds are present, which prolongs the effects of drug. The effect continues until the drug is excreted or a new receptor is generated. Prolonged contact of tissues with the agonist results in decreased number of receptors in the tissues called downregulation of receptors. Examples include the patient suffering from bronchial asthma, in whom by prolonged usage of beta agonist downregulation occurs. This effect is reduced. Now coming to the another portion, that is dose-response relationship. The characteristics of exposure to a chemical and the spectrum of effects caused by the chemical come together in a correlative relationship that toxicologists call the dose-response relationship. This relationship is the most fundamental and pervasive concept in toxicology. To understand the potential hazard of a specific chemical, toxicologists must know both the type of effect it produces and the amount or dose required to produce that effect. The relationship of dose to response can be illustrated by a graph called as a dose-response curve. There are two types of dose responses. First one is graded dose response and second one is quantile dose response. Graded dose response describes the graded response of an individual to varying doses of the chemical, and quantile dose response describes the distribution of responses to different doses in a population of individuals. An important aspect of dose-response relationships is the concept of threshold. For most types of toxic responses, there is a dose called a threshold, below which there are no adverse effects from exposure to the chemical. The human body has defenses against many toxic agents. Cells in human organs, especially in the liver and kidneys, break down chemicals into non-toxic substances that can be eliminated from the body in urine and feces. In this way, the human body can take some toxic insult that's at a dose that is below the threshold and still remain healthy. The identification of the threshold beyond which the human body cannot remain healthy depends on the type of response that is measured and can vary depending on the individual being tested. Thresholds based on acute responses such as death are more easily determined, while thresholds for chemicals that causes cancer or other chronic responses are harder to determine. Even so, it's important for toxicology to identify a level of exposure in a chemical at which there is no effect and to determine thresholds when possible. A comparison of dose-response curves among chemicals can offer information about the chemicals as well. A steep curve that begins to climb even at a small dose suggests a chemical of high potency. The potency of a chemical is a measure of its strength as a poison compared with other chemicals. The more potent of the chemical, the less it takes to kill. Now, the drug potency and its efficacy. The content of drug that is required in order to produce a given percentage of its highest effect regardless of the magnitude of maximal effect. A drug may have higher potency but least efficacy, means the response is seen at very lower doses and remains very small even at higher doses. Drug potency is sometimes an important clinical consideration. Drug potency depends on both affinity and efficacy of the drug. Potency is that dose of the drug that is required to produce a specific effect of the given intensity when compared to a standard reference of a drug. Potency is a measure of the drug activity expressed in terms of amount that is required in order to produce an effect of the given intensity. A highly potent drug, for instance, for example, morphine, alprazolam, and chlorpromazine gives a large response at low concentrations. When compared to a drug of lower potency, for instance, ibuprofen, ethyl salicylic acid, evokes a small response at low concentrations. It's proportional to affinity and efficacy. Efficacy is the relationship between the receptor occupancy and ability to start a response at molecular, cellular, tissue or system level in the body. The response is equal to effect and depends on both the drug binding and the drug bound receptor, thereby producing a response. Hence, potency depends on both affinity as well as efficacy. Now, the therapeutic index. Actually, the therapeutic index, also referred as therapeutic ratio, is a comparison of the amount of a therapeutic agent that causes the therapeutic effect to the amount that causes toxicity. The related terms, therapeutic window or safety window, refer to a range of doses which optimize between efficacy and toxicity, achieving the greatest therapeutic benefit without resulting in unacceptable side effects or toxicity. Classically, in an established clinical indication setting of an approved drug, TI refers to the ratio of the dose of the drug that causes adverse effects at an incidence or severity not compatible with the targeted indication. For example, toxic dose in 50% of subjects TD 50 to the dose that leads to the desired pharmacological effect. For example, efficacious dose in 50% of subjects ED 50. In contrast, in a drug development setting, TI is calculated based on the plasma exposure levels. In the early steps of pharmaceutical toxicology, TI was frequently determined in animals as lethal dose of a drug for 50% of the population, that is LD50, divided by the minimum effective dose for 50% of the population, that is ED50. Today, more sophisticated toxicity endpoints are used. A high therapeutic index is preferable for a drug to have a favorable safety profile. At early discovery or development stage, the clinical TI of a drug candidate is not known. However, understanding the preliminary TI of a drug candidate is of utmost importance as early as possible since TI is an important indicator of the probability of the successful development of a drug. Recognizing drug candidates with potentially suboptimal TI at earliest possible stage helps to initiate mitigation or potentially redeploy resources. In a drug development setting, TI is the quantitative relationship between efficacy and safety, without considering the nature of pharmacological or toxicological endpoints themselves. However, convert a calculated TI to something that is more than just a number. The nature and limitations of pharmacological or toxicological endpoints must be considered. With this, we come to the end of the lecture. Hope